So our first speaker will be SCI researcher Peggy Assinck. Am I saying that right?
Yeah.
Should have asked first. Peggy studies cell transplantation for SCI at ICORD, the International Collaboration on Repair Discoveries. She has received several prestigious fellowships, including the Canadian Institutes of Health Research, Frederick Banting and Charles Best Canada Graduate Scholarship. She has also played sledge hockey for 19 years and has been on the Canadian Women's National Sledge Hockey Team for 6 years. Welcome, Peggy.
Thank you. All right. Can everyone hear me at the back, or would you rather if I used a mic? Mic? We're all mic? Okay, perfect. I only have so many hands, so we'll see how this goes. Okay, can everyone hear me? Okay, perfect. We're good? Okay. So my name is Peggy, and I am a PhD researcher at ICORD. And my goal today is just to talk a little bit about the biology of spinal cord injury and why cell transplantation might be a useful thing. I'm also going to touch on some research that was done recently that kind of caught the attention of the media, looking at cells that you can isolate that are important for our ability to smell and then transplant those cells back into the injured spinal cord. And I'm going to finish up by talking a bit about my research and what happens here in Vancouver and at ICORD. So to start off, what I'd like to do is talk a bit about the spinal cord. So this is an example of a spinal cord. This does not have an injury as of yet. And what I'd like you to notice is these long blue lines, and what they represent is axons. And these axons carry information up and down our spinal cord. So they carry information from my brain to my big toe to tell it to move, or sensory information from my big toe back up to my brain. And they can also be associated with autonomic functions like bladder function, bowel function, sexual function, etc. And so carrying this information up and down the spinal cord is really important. Now, in and around these long blue axons, you can also see these other cell types. And there's lots of other cells in the spinal cord, and they contribute to lots of important functions. Now, what happens often after spinal cord injury is we get a loss of many of these cell types. So this is an example of an injured spinal cord. And what you can see is this large fluid-filled cyst, and it's void of a lot of those cells that used to be there. And importantly, you also see these axons. Let me see if I can get these pointer to work. Nope, good try. So you can also see these axons that used to be going up and down the spinal cord that were communicating information, that many of them have actually been severed. And therefore, it stops some of the communication from above and below the injury site. Now, really importantly, depending on the severity of the injury, you can get different amounts of damage. So in this situation, you've got a large amount of damage in the middle, but on the two outside, you can see that there is still some axons that may be able to communicate some information down the spinal cord. And that's very much dependent on each and every spinal cord injury. Okay? Now, when we think about the problems and the biology behind spinal cord injury, this is kind of where researchers got this idea of cell transplantation. And so the idea is that if the major problem after injury is that we get the loss of many cells, then maybe transplanting a cell into that area would be beneficial. So that's kind of the basis of what we're talking about today. Now, the idea would be that these cells would have the ability to fill in some of this area, this large fluid-filled cyst, and they'd be able to differentiate into different types of cells that could be beneficial. Okay? Now, what I'd like to point out is in a perfect world, those cells would go in and they would allow for axons to regrow across and make new connections, and these cells would be able to become all those important cell types that were there before. Now, unfortunately, at this point, we're not living in a perfect world, and so there's a ton of research that needs to happen in order to understand how these cells become different cell types, how they integrate, whether they survive, and that's kind of a lot of the work that's being done right now. All right, so the next thing that I'd like to do is talk a little bit about cell sources. And so the idea is that we can grow cells in a dish and then transplant them into the injured spinal cord. But where do we get these cells? And there's a bunch of different answers to that researchers have been interested in for a long time. But one thing that I'm sure many of you guys have heard about is stem cells, right? And so stem cells is the ability of a single cell to become multiple different cell types. So for example, if I've got a cell here, it could become a muscle cell and a hair cell and a bone cell, okay? And that makes it a stem cell because it has the ability to differentiate into lots of different cells, okay? Now, those might be ideal in this situation because they could help to become the different cell types that we're looking for. But there's also another group of researchers that are looking closer at looking closer at other types of cells. So stem cells we can isolate from the embryo, we can isolate them from the fetus, and we can also isolate them from different tissues within the adult body. Okay? Now, the other source of cells is not stem cells. So these are cells that are in our body all the time, and if we take them out of our body, grow them in a dish, and then transplant them in the same form that they were before into the injury site, they may be beneficial. And that's what I'm actually going to talk a little bit about now. And so I'm going to talk about, and I think there's some information on all the tables about this, but I'm going to talk about a study that the media got very excited about a couple months back, and maybe some of you have heard about it. And essentially what they did is they took a cell type that is important for our ability to smell. So it's actually isolated from the brain, but it's an important cell for our ability to just be able to smell. And what happened very quickly, and I'm going to show you some clips of this, is that there was a video that was put out by BBC originally, and it got picked up by lots and lots of different media organizations throughout Canada. And there was these great headlines, and one of the biggest ones was nasal cells bring major breakthrough for spinal cord injury. And there was also other, a little bit more modest headlines that read, spinal cord injury, big hope is cell therapy, but exercise is the best for now. And so what I'd like to do is actually show you one of these video clips. And so this is a clip that is shown in CTV. And it's actually featuring my supervisor, Wolfram Tetzlaff, who's going to come up here soon. But I want to let you guys kind of see what it is that was portrayed in the media specific to this study. So here you go.
These remarkable images show Derek Fidiker after a pioneering cell transplant. He'd been paralysed from the chest down, but now movement and sensation are slowly returning to his lower body.
Cars are running down.
Scientists used cells from one of his olfactory bulbs, shown in green, part of the sense of smell, because nerve fibres there constantly regenerate. Surgeons in Poland injected the cells into the gap in his damaged spinal cord to stimulate repair and regrowth. The results, published in the journal Cell Transplantation, have astounded the lead UK scientist.
You are making history now. To me, this is more impressive than a man walking on the moon.
Okay, so what I'd like to do and what my goal is now is I've now shown you kind of what was portrayed in the media about this story, and what I'd like to do is talk a little bit more about the science, and I'm sure that this discussion will go on throughout the night with regards to bringing up our speakers who are experts in the field. But what I want to talk about is the important part about this study is that this is essentially a case study. And so what that means is that this was one gentleman who received a procedure, and this is the outcome of that one procedure. So I want you to keep that in mind as we go through this. All right, so the neat thing about this is that this case study was actually published in a paper, so this paper, the Journal of Cell Transplantation. And so we have lots of information about what happened. And so I'm going to go through the details in a little bit more detail. So there was 2 procedures that were done on this gentleman. The first one was to remove one of his olfactory bulbs. And so again, that's a piece of the brain that's important. It contains cells that are important for our ability to smell. And so he essentially got one of those olfactory bulbs removed. They grew it in a small dish for about 2 weeks. And when the cells were ready, they then did a subsequent transplantation surgery. And this was kind of the big surgery. And I want to take you through what it is that happened in those moments. So that major surgery kind of had 3 different parts. The first part was that they needed to find the injury site. And the researchers and the surgeons reported that in order to find the injury site, they actually needed to untether the spinal cord. And what that means is that in response to an injury or different situations, you can get a lot of scar tissue, and that's really, really dense tissue. And it can build up in and around the spinal cord. And this tissue can actually contribute to deficit, so it can actually squeeze some of these nerves and block some of the impulses in and around an injury site. And I can actually attest to that quite well, because although I have spina bifida, which is the reason that I originally had some scar tissue buildup on my spinal cord, but I've actually had about 6 surgeries in order to repair just tethered scar tissue on my spinal cord. And it's allowed me every time I've gotten the surgery to be able to walk a little bit better. So untethering of the cord is something that sometimes for certain people can result in functional improvement. So I want to note that. That's the first thing. The next thing they did is once they found this injury site, in this example, the circle number 2, what they noticed is that this spinal cord, which is the large blue line that you can see here, you can see on one side of it there's an 8mm gap. And you can see on the other side there's still a little bit of intact tissue, and that's something I want to point out. And what they did is they did many, many small injections of these cells above and below the injury site, over 100 injections of these cells that they isolated from the olfactory bulb. Now, the next thing that they did is they did a small incision in his ankle, and they took out a small nerve segment, and they cut it into 4 pieces and laid those strips across this large opening. And so putting these 3 things together, the surgery that he underwent untethered the spinal cord, injected these cells in and around the site, and put these nerve fibers to help it bridge this site. Okay, so those are the details you need to know with regards to the procedure. The next thing I want to talk about is the timeline of this case study. So important details that you need to know. This gentleman had had an injury for 13 months before he had any contact with the researchers. When the study started, what he did is underwent extensive rehabilitation. So for 8 months pre-transplant, he did 5 hours of rehab each and every day for 5 days a week for an entire 8 months before he even did his surgery. Okay? And then he did 2 years or 24 months worth of that same intensive rehabilitation program after his transplant. So this is another really important factor that we need to be aware of. Now let's talk about some of the improvements that we're seeing. So at about 3 months post-transplant, so that big green arrow is meant to be that big transplant surgery. About 3 months post-transplant, he started to get some movement in his left thigh that he didn't previously have. Okay, an ability to fire that muscle. About 6 months after the injury, he reported, well, not just he reported, researchers reported his ability to walk using bracing. So this is kind of the picture on the bottom left. So he's got full braces on both of his left and his right leg, and is using weight support on these parallel bars in order to help him to move across the ground. Now, at 2 years following the transplant, what he was able to do is walk around independently outside, and that's pretty exciting. And what he did in this picture that you can see here is he's still got some help from bracing. He's still using a walker that's supporting some of his weight, but he is able to do some movements that were much more than what he ever was able to do pre-transplant. Okay? And so this is the details that I want you guys to know and think about. And moving forward, what I want you to be aware of is just that this is a single patient or a single person who underwent a procedure that had some positive outcomes. And that's something that I think we're going to talk about a little bit more moving forward with our panel discussion. So next, what I'd like to do is talk a little bit about what's happening in Vancouver. So I'm a PhD student. My role is looking at stem cell therapies after spinal cord injury. Now, most of my research, actually all of my research, involves experimental work. And so I'm interested in how it is that cell transplantation could be beneficial, but I always look in a rodent model. And so I'm not yet doing transplantations in humans or larger animal models. And what my work has shown is that if you take a stem-like cell that you can isolate from the skin, and you grow it in a dish, and you push it towards a cell type that you think is pro-reparative, something that's going to be beneficial to put in the cord, and then transplant those cells into the injured spinal cord, what we see, and this is actually an image from a rat spinal cord, and you can see these green cells have survived quite nicely. And you can see these red axons, it's kind of diagrammed up at the top, as well as, I don't know if you can see it super well here, but there are some axons that are able to grow into these transplanted cells. Now, importantly, when we ask the question, do animals that receive this transplant, did they do better than animals that didn't? The answer is there is evidence that there is small improvements. So not big improvements, but small improvements from doing this cell transplantation work. And so the rest of my work and kind of what we're doing at ICORD is to try and figure out how we can improve upon this. And this is a really important aspect of work that's done before we would want to move towards trying this in larger animal models or humans. Okay? The last thing that I want to quickly do is just tell you about where you can get more information. So one thing is the ICORD website, and this blog is actually out on your tables, so there's lots of information with regards to up-and-coming research that's happening. Also, this website is a really great website. It's www.closerlookatstemcells.org, and this is great information for people who are thinking about stem cell therapies or cell transplantation work. And also there is a website called clinicaltrial.gov, and this is run through the NIH, and you can actually do a search for something like spinal cord injury and get information on trials that are registering with this website. So just a way to access different bits of information. Okay? And the last thing I'm going to leave you with and wrap up with is a short video, and this video was put together by the Stem Cell Network. And what it is focusing on is a thing called stem cell tourism. So that's the idea, actually it's going to explain it really nicely, but it's the idea that that there might be treatment options in other parts of the world that look quite promising. And so when we talk about cell transplantation or stem cells, I kind of think that they go hand in hand. So I think they both apply here. So I'm going to finish off with this quick video. And thank you so much for listening. And
in the past 50 years, stem cell research has led to some groundbreaking therapies, including bone marrow transplantation and skin grafting. Today, stem cell research continues to hold the promise of new treatments for many diseases. However, most of these potential treatments are still experimental. Any experimental therapy must first be carefully tested to make sure it is both safe and effective before it is approved for patients. This testing process normally takes several years, during which time many experimental treatments may fail because they are unsafe, ineffective, or both. Despite this, clinics throughout the world offer untested and potentially dangerous stem cell-based treatments, usually at a high cost to the patient. As a result, these locations have become a destination for stem cell tourism, where hopeful patients travel internationally to seek unapproved stem cell therapies. Before patients spend time and money, they should familiarize themselves with the facts and think twice about obtaining expensive treatments that have not yet been proven safe or effective.